It's a pleasure to introduce Rob Eagle today to what may or may not be our first sort of joint seminar between the department on campus responsible for the fluids on planets, atmospheric and ocean science, and ecology and evolutionary biology. And one of the reasons why we're doing this is that Rob is a target of opportunity, and so we're trying to look around the mosaic of academic students at UCLA to figure out where he might possibly fit in. So Rob has got his undergraduate degree at Durham University, and then went on to a named studentship at Cambridge, where he got his PhD in 2005. And he was working then on cancer and loosely what goes on the surface of cells in cancer. But by 2008, he had a Chancellor's Fellowship at Caltech and was on to looking at sort of geobiological problems that involve biomineralization. Some of the more noteworthy of those are his work on trying to figure out what dinosaurs were up to physiologically with the temperature of their bodies and so forth. And he's done a variety of other things. He was involved in a floating university that convened somewhere off the Antarctic Peninsula. And he continues to work on a variety of questions that involve the paleoclimate of the Antarctic and the Arctic. But he's also sort of now going on and working on not the past, but what you can look at with the future, or relative to our future of the ocean with biomineralization. And so he's now doing research both on two continents, both here at UCLA and at the European Marine Institute in France. And so on that note, I'd like to also note that his title is attractive enough to induce some people to come up from Orange County. So we have some people here from Chapman University, as well as people from the two departments aforementioned. So without further ado, we have Rob's title up here, and I'll let him proceed.
Well, thanks for the kind introduction, Dave. So, what I'd like to talk to you about today is really a series of research projects that my group's developed over the last couple of years that are really focused on trying to understand the response of marine organisms, and in particular marine organisms that produce calcium carbonate shells or skeletons to a high CO2 world, and in particular CO2-induced ocean acidification. This group's a kind of Franco-US collaboration at the moment, involving researchers both here at UCLA, but also in France, where I also have an appointment. So to give you an idea of the structure of my talk today, I'll spend a bit of time introducing the problem of CO2-induced ocean acidification and its potential impacts. I'll then go on to talk about some of the work that we're pursuing at the moment, which includes developing a kind of conceptual model to try and understand how organisms respond to OA. And then I'll talk about some experimental data that we're collecting to try and test those models. And the experimental data involves both geochemical measurements, but also approaches from cellular biology as well. And then I'll go on to talk about some work that we're just in the process of setting up at the moment, and we're hoping will form a component of our research program going forward, which is really to design experiments to try and look at the long-term impacts of ocean acidification on marine organisms in kind of long-term experiments that allow for organisms' ability to adapt potentially to environmental change. So as I'm sure most people here are aware, atmospheric CO2 levels are rising due to anthropogenic emissions, here shown in a kind of classic time series from the Mauna Loa Observatory. And the oceans are a significant sink of atmospheric CO2, and you can see this from a nearby oceanographic station a corresponding increase in the concentration of dissolved CO2 in the upper ocean. This increase in dissolved CO2 is also accompanied by a corresponding decrease in oceanic pH. And the reason for this is linked to the oceanic carbon cycle. So when CO2 is taken up into the ocean, it undergoes a hydration reaction with water to produce the two dominant forms of dissolved inorganic carbon in the ocean, bicarbonate ion and carbonate ion. These hydration reactions produce protons, which is the reason for the pH decrease. This plot here is called a germ plot, and it simply shows the concentration of the three main dissolved inorganic carbon species in seawater as a function of pH. And at the pH of typical ocean surface waters, you can see that bicarbonate is significantly the dominant form of dissolved inorganic carbon compared to carbonate ion. So how does this relate to organisms that precipitate calcium carbonate shells? Well, it's thought that most organisms that do this, like corals or calcifying phytoplankton, for example, predominantly use carbonate ion as the source of ions to precipitate their calcium carbonate biominerals. And therefore, they might be quite sensitive to changes in the concentration of this dissolved inorganic carbon species in seawater. It's considered kind of useful in this field to consider a parameter called the saturation state of seawater. There are two different saturation states that are relevant for biomineralization studies. The aragonite and calcite saturation state. These are the two main polymorphs of calcium carbonate produced in the ocean. This is a definition of this parameter, but the main thing to remember really is that a saturation state of less than 1 is the point where this mineral will start to dissolve in solution. And aragonite saturation states of less than 1 are indicated in these two maps by a kind of red colour. And you can see in a hindcast of aragonite saturation states of ocean surface waters from pre-industrial times, so almost none of the upper ocean is undersaturated with respect to aragonite. However, in a future projection, assuming a kind of business as usual CO2 emission scenario, you can see that large areas of the polar oceans have this red colour and predicted to be undersaturated with respect to aragonite. In other words, that mineral would dissolve in seawater by the end of this century. There's also a corresponding decrease in saturation states in most areas of the ocean. So what impact are these changes likely to have on marine calcifying organisms? This is a kind of typical experiment. There's lots and lots of these type of experiments being published now, examining a kind of coral reef ecosystem that's been grown in a controlled environment, and CO2 has been varied, causing a decrease in carbonate ion concentration. On the y-axis is calcification rate of the coral in this experiment. And you can see that there's in this experiment at least a fairly linear decrease in the calcification rate of corals as CO2 is increased, even up to a point where there's actually net dissolution or dissolving of calcium carbonate at these high CO2 conditions. I just want to highlight at this point, though, that the impacts of ocean acidification aren't just linked to calcium carbonate biomineralizing organisms. There's growing literature of the impacts of OA on a broad range of different marine life, from cyanobacteria all the way up to large vertebrates like fish. It's been found that OA and changes to the carbonate system can cause a variety of ionic imbalances in the tissues of fish, including kind of hyperpolarization of neurons and activation of GABA receptors, which has been found to alter the behavior of some organisms, both their migratory and homing behavior, and also their response to things like predator cues. So I just put this slide up here to emphasize that the potential impacts of OA on marine life are really quite broad. So in response to a major environmental perturbation like ocean acidification, organisms have a few different responses available to them. They can obviously move or migrate away from that environment into a more favourable environment if they're able to. Some organisms may just be refractory to the environmental change, or they may have mechanisms already that allow them to acclimatise to those change. Or they might be able to evolve, effectively adapt to the changing environment. This is potentially quite difficult for some species due to the rapidity of global change that is likely to be occurring over the next century. But it's certainly possible for some species that they may be able to do this. Of course, if a marine organism is not able to do this in response to OA, it could face an eventual extinction. Oops, sorry. And it's a fairly obvious point worth making nonetheless, that a negative impact of ocean acidification on a single species is not just going to have an effect limited to the abundance and health of that one species, but there's going to be significant knock-on effects on oceanic food webs and ecosystems. And it's also important to highlight that the impacts of ocean acidification are not just limited, not just of concern to those of us who are interested in the welfare of marine life, but they're also relevant to those of us who are interested in global biogeochemical cycles and the role of life in the Earth system. So, for example, this calcifying phytoplankton, Emiliani huxleyi, is microscopic in size, but it actually accounts for a very large proportion of primary productivity in the ocean, up to 20% in some regions, and it forms these enormous blooms, such as this one that can be seen from space off the coast of Newfoundland. And so potentially impacts of ocean acidification on organisms like this can have an impact on kind of globally significant biogeochemical cycles. So this slide shows a kind of complex diversity of biogeochemical cycles. One thing, many of which could be potentially impacted by ocean acidification. One thing I want to highlight, though, is that primary productivity and the abundance of things like Emiliani huxleyi and their formation of blooms in the upper ocean involved in the biological carbon pump, which is basically a mechanism whereby organisms fix carbon in the upper ocean, and then when they die they'll fall into the deep ocean, effectively exporting carbon from the upper ocean to the deep ocean, where it can be sequestered for a period of time. And global carbon cycle models that seek to predict the response of the carbon cycle and climate system to future change often parameterize the biological carbon pump, but they do so in kind of the terms of physical oceanography. So they might say, for example, well, future change will increase the stratification of the ocean, which may lead to carbon being stored for a longer period of time in the deep ocean. But what these models don't tend to do is actually take into account the potential impact of things like ocean acidification on the biology of these organisms. So what if OA reduces the abundance of primary producers in the upper ocean? And so maybe this is an area where biologists and biological experiments can provide some constraints to carbon cycle models and biogeochemical models. There are other potential biogeochemical cycles that are also impacted in OA. There's not really time to go through all of them. Some implications that OA may reduce nitrification rates in the ocean by impacting the activity of cyanobacteria. Impacts on the behavior and migration of things like fish and zooplankton may have impacts on processes like the biological mixing of ocean waters. There's really quite a broad range of potential impacts of OA on life that will be passed on to biogeochemical cycles and physical processes in the ocean. Another example I'd like to highlight is that coccolithophore blooms in the ocean are known to be a major source of dimethyl sulfide to the atmosphere. So when these blooms form and they senesce or die, DMS is released into the atmosphere, and so these phytoplankton blooms play a role in the sulfur cycle as well. And DMS is also known to be a significant aerosol that is involved in cloud formation. Therefore, the abundance and kind of health of these phytoplankton blooms potentially has feedbacks to atmospheric processes as well. So,
with aerosol, it was very interesting, but I was just wondering whether some evidence of potential effects. Is there any specific evidence that some aerosol is generated in the atmosphere?
yeah, I mean, I think it's well established that these phytoplankton blooms, they contain precursors of DMS, and when they die, they're kind of released into the atmosphere. I think that's fairly well understood. The effects on OA on that process are not well understood. So there's some studies that have been done on kind of mesocosm culture experiments, which are, experiments where you culture a community of phytoplankton, and they've shown that in acidified seawater there's a change in the amount of DMS production in these mesocosm experiments. So there's a study cited there where that's been done. I think this system, though, and its response to OA is kind of quite unconstrained at the moment. So we've previously seen an experiment where we saw a kind of negative impact of high CO2 and ocean acidification on coral biomineralization in a controlled experiment. Is there evidence out there in a kind of Earth system scale that we can actually observe potential impacts of acidification? So this is a study that was published very recently in 2015, and it's a satellite-based observation study where they were looking at the rates of inorganic carbon production or calcification in the Southern Ocean over the last 16 years. And basin-wide, they observed about a 4% reduction in calcification over this time period, with the regions of the Southern Ocean highlighted in blue here of up to 10% reduction. So in some localized regions, up to 10% reduction in calcification, also with some areas of increase there. What's kind of interesting about this study is that they didn't see any change in chlorophyll content, suggesting that there was no change in the abundance of photosynthetic algae in the upper ocean, but there was a reduction in calcification, and that might indicate perhaps a kind of change in community structure between calcifying and non-calcifying phytoplankton in the Southern Ocean. So a kind of unconstrained observation in a way, but an indication that potentially kind of global scale changes are underway in marine calcification. So bringing this back to my research, our current research projects are really to try and understand how changes in the physical environment might influence biological processes like marine calcification, with a specific goal of trying to understand which marine organisms are going to be able to acclimatize and do well and survive changes like ocean acidification, and but also understand which species may not do so well. And we're also kind of interested in what happens when you consider genetic variability and adaptive responses, and also with an eye on how these experiments can perhaps provide some constraints or useful information to people interested in biogeochemical cycles and modeling. So kind of the most fundamental question really is, do all marine organisms respond in a negative manner to ocean acidification? And one of the most important early pieces of work addressing this question was by my close collaborator Justin Rees, who did a kind of heroic culturing experiment where he took 18 different marine organisms, cultured them all in the same conditions at variable CO2 and characterized their responses. This was published about 5 or 6 years ago. So he did cultures at 400, 600, 900 or 2850 ppm CO2. This kind of busy slide is shown for a reason, because I want to highlight the diversity of responses. So on the x-axis here we've got aragonite saturation state, which decreases from right to left up to undersaturated conditions right on the left here. And on the y-axis you've got calcification rate. So you've got the production of calcium carbonate on the left, which decreases from the top to the bottom. And the first thing to note is that a variety of these marine organisms at the bottom here, typically kind of mollusks, showed a negative response to elevated CO2 conditions. Many of them showed what you describe as a kind of linear negative response. So the calcification decreased as CO2 increased. There's a bunch of organisms in the middle here, including some corals, which showed resilience to elevated CO2 conditions until they reached a certain point. So the very high CO2 conditions, they showed a drop in calcification. And there were some organisms that were basically unaffected. It was kind of interesting, and one reason this study was particularly important was that there were a collection of organisms at the top here, including some echinoids and crustaceans, actually showed positive growth responses to elevated CO2 conditions. In other words, they produced more calcium carbonate at high CO2 than they did at low CO2. A kind of surprising and interesting result. And so this schematic kind of summarizes the variety of different responses seen in those 18 organisms, including the kind of most predictable, perhaps negative response. Also parabolic responses where lower CO2 increases, there was an increase in calcification, but then it dropped back down again at very high CO2 threshold responses, but also these positive growth responses.
Could you just quickly map out how these changes in aragonite saturation map on to changes in global CO2? So we have a sense of how this scales with the probable changes in CO2.
So in this experiment, it was only the 2850 treatment that was actually undersaturated with respect to aragonite. And these were all done at 25 degrees C, which is part of the reason for that. So it was only the 2850 that was undersaturated with respect to aragonite.
So 0.5 to 2.5 corresponds to 2050 to 400?
So this is, yeah, 400, 600, 900. Excuse me. So the question is, how can we explain this kind of remarkable diversity in responses to high CO2 conditions in marine organisms? And the explanation I'm going to spend most time on for the rest of the talk is relates to trying to understand how different organisms might have different capacities to regulate their internal pH, effectively regulating it and buffering it from changes in pH in the carbonate system of seawater. So that's what I'll spend a lot of time on for the rest of the talk. There are other potential explanations that are also relevant though. So there are potential linkages between the internal carbonate system of organisms to other physiological processes, such as photosynthesis, that need to be taken into account. There's variability between organisms in the type of organic materials that they might produce. Some organisms may produce kind of organic layers to protect their shells or skeletons from undersaturated or corrosive seawater. And there's also, as we touched on before, a variability in the type of calcium carbonate polymorph that organisms produce, with some, for example, aragonite, more prone to dissolution than calcite. So now we'll move on to our work, and I'll first present a sort of conceptual model that seeks to explain this diversity in response to ocean acidification. And then I'll go on to some of the data we're collecting to try and test that model. And so this model really relies on trying to understand how organisms might regulate the proton abundance or the pH of their internal calcifying fluid relative to that of seawater. So this is an example of a hypothetical growing coral. And so in this example we have a growing coral skeleton here. It's bathed in a fluid which is the ultimate source of ions to the growing skeleton, and it's also enclosed in tissue, so it's not directly exposed to seawater. However, as indicated in this slide, seawater must ultimately be able to access the calcifying fluid, because that is the source of dissolved inorganic carbon for the growing skeleton. And it's debated in corals and other organisms exactly how seawater and DIC is transported into the calcifying fluid. Is there direct seawater leakage, so there's effectively no barrier between seawater and this fluid? Does it diffuse across membranes, or is there a very sort of active and controlled biological process to transport seawater and DIC to the calcifying fluid? That's kind of unknown and probably varies a lot between different organisms. And the second part of this relates to organisms' ability to transport protons out of the calcifying fluid. Do they have transporters that are able to remove protons from the calcifying fluid, changing the pH and carbonate system of this fluid with respect to seawater? So why would they want to do this? Well, if an organism can pump away protons from the calcifying fluid, it can elevate the pH of its calcifying fluid with respect to seawater, which increases the abundance of carbonate ion in the calcifying fluid, which promotes its combination with calcium ions and promotes the precipitation of a calcium carbonate shell. So it's potentially advantageous for organisms to do this. Whether this proton pumping exists in different species is uncertain, and the exact molecular basis for the proton pumping is not known either. So it is a kind of hypothetical thing to some extent, although I'll show you some evidence that it also exists later. So to take this conceptual model further, so we'll return to this germ plot showing the abundance of dissolved inorganic carbon species as a function of pH, initially at a kind of modern level of CO2. So an average pH of ocean surface water of 8.16, you'll get around 182 micromoles per kilogram of carbonate ion, but with bicarbonate ion being the dominant form of DIC. So in this conceptual model, for example, if we consider a coral perhaps that has a moderate ability to pump away protons from its internal calcifying fluid, this will effectively have the effect of tripling or more the abundance of carbonate ion in its calcifying fluid if it pumps away 100 micromolar of protons from its calcifying fluid, thereby making the precipitation of its calcium carbonate shell more favourable. But now if we move to a kind of hypothetical high CO2 scenario, you're going to see acidification of the surrounding seawater, which is going to cause a drop in the carbonate ion concentration. But you're also going to see an overall shift upwards in the these curves of the Bjerrum plot, simply because you're adding a lot of carbon into this system. You're increasing the amount of dissolved inorganic carbon in solution. So now a coral with a kind of moderate ability to remove protons from its site of calcification will only get up to around 400 micromole per kilogram. So it has less carbonate ion present at its site of calcification, and that may inhibit the production of the coral skeleton. But now let's consider another scenario. So we have a high CO2 scenario and a reduction in carbonate ion concentrations in seawater as a result. But now we're going to consider an organism that has a very strong proton pumping capacity, can pump a lot more protons out of its calcifying fluid, has a strong control over its internal carbonate chemistry as a result. Maybe it can elevate the pH of its calcifying fluid to over 10, and here it's generating a very high concentration of carbonate ion, which will really sort of drive the precipitation of calcium carbonate biominerals. What's interesting about this scenario is that in the high CO2 scenario, because of the input of dissolved inorganic carbon into this system, there's actually more carbonate ion produced at the site of calcification in a high CO2 scenario than there is at a low CO2 scenario. So here's the low CO2 scenario producing 1600 micromoles per kilogram, and the high CO2 scenario producing 1800 micromoles per kilogram. So this is another way of looking at this type of response. So here we have calcification site pH and the abundance of ions on the y-axis here. And so here we're looking at carbonate ion concentration in a hypothesized organism that has a strong proton pumping capacity. Here we have 400 ppm, 600, 900, and 2850 ppm CO2. In this organism, there's a consistent increase in carbonate ion concentration at the site of calcification as CO2 increases. And you can potentially see how that could be an explanation for some of these positive calcification and growth responses to high CO2 conditions. There's just more carbonate ion and dissolved inorganic carbon available to these organisms to grow. In this kind of mid-range example, you can see how you might be able to generate a parabolic response where you have an initial increase in carbonate ion concentration as CO2 increases, but then a drop-off in a kind of moderate proton pumping organism. That's something that was observed in Justin's experiments. But if we have an organism that only has a weak capacity to regulate pH at the site of calcification, you get a consistent drop in carbonate ion concentration at the site of calcification, and maybe that is associated with these linear negative responses. So why might organisms differ in their ability to regulate pH at the site of calcification? Well, the main reason is probably that there's a significant energetic cost associated with maintaining these proton gradients. And so maybe organisms like tropical surface corals, for example, that grow in environments with supersaturated waters where there's plenty of carbonate ion available, don't really need to expend this energetic cost. But other examples like deep sea corals, for example, that grow in deep waters that are potentially more undersaturated, they may well have evolved mechanisms to regulate their internal calcifying fluid pH. So the energetic cost with maintaining these ion gradients can be described by the Nernst potential, and it relies on the ratio of external protons to internal protons. And exactly how organisms regulate that is a kind of key uncertainty in this conceptual model. So there are potentially two end-member scenarios. One, that an organism maintains a fixed concentration of protons pumped across a membrane as CO2 changes. Alternatively, there might be a fixed ratio maintained between seawater and their calcifying fluid. And we have no way from this model of really knowing which is most appropriate. In the examples I just took you through, we were assuming this was the case, but both of these scenarios are equally plausible. And so what we really need is some experimental data to test some of the predictions of this conceptual model that I just put forward. And there's a variety of kind of approaches and techniques available to us that we can potentially use. pH microelectrodes is one technique. Effectively, that's a very tiny pH probe that you can insert into organisms, into what you think might be their calcifying fluid, and to measure the pH of that. pH-sensitive dyes is a similar idea. You take a section of an organism and introduce this dye, which has a colorimetric output relating to the pH of the solution. Another one that we're pursuing in particular is looking at the geochemistry of the carbonate produced. And the idea here is that the chemistry of carbonates is quite complex, and there are aspects of it that can be measured and probed that can be directly related back to the pH and carbonate chemistry of the solution that the carbonate grew from. And the tool I'll spend most time talking about in this lecture is boron isotopes, but I just want to highlight that there's a whole variety of other geochemical measurements in our toolkit that can also be applied to these type of questions. So as part of one of our projects, this is some preliminary data we've generated using the microelectrode approach. This is kind of how a microelectrode might be inserted into a growing coral. And in this particular species, we observed that the calcifying fluid, as recorded by this microelectrode, the species had a pH of over 10, which is 2 pH units elevated with respect to seawater, suggesting that this species has quite a strong control over its internal pH. When this coral was taken from ambient seawater and placed in acidified seawater, we saw a big crash in the pH of its calcifying fluid, followed by a recovery over a course of about 3 hours. And it kind of plateaued out at a point, but did not recover to the original pH. And so here are some of the parameters determined from that experiment. So in ambient seawater, the pH of the calcifying fluid was about 2 pH units higher than that of seawater, leading to a highly saturated calcifying fluid and much higher carbonate ion concentrations. In the acidified scenario, we saw a drop in the pH of the calcifying fluid, a drop in saturation state and carbonate ion availability. But interestingly enough, the ratio of external to internal protons remained the same. In other words, this coral hadn't developed an adaptive response. It wasn't trying to compensate for the external change in pH. It was still maintaining the same ratio. There are various criticisms of these microelectrode approach and also the pH-sensitive dye approach that are discussed in the community that works on these kind of things. Namely, well, firstly, does the insertion of these kind of electrodes cause stress and damage to the organism? One of the main ones really is, are you actually sampling the correct calcifying fluid? at least if you talk to geochemists, what's really important is the chemistry of the fluid right at the interface of the growing mineral, at the point where it's growing. So if you're sampling fluid somewhere away from that, is that really representative? They're also kind of short-term observations, so that experiment I just showed you was only over 3 hours, but what if you grow your marine organism in a acidified environment over a course of a month or 6 months or something? Will the response that you're observing still be the same? So this is where we turn to geochemistry. So if we measure the geochemistry of the carbonate, firstly we know that obviously that reflects the exact chemistry of the calcifying fluid that the carbonate was formed from. And you can also make measurements, say, on material that's generated over long timescale experiments. So how does this boron isotope approach actually work? So boron in seawater exists in 2 main forms, boric acid and borate, and their abundance varies according to pH. So that's that relationship. These 2 forms of boric acid and borate have a very different boron isotope composition. And crucially, marine carbonates, indicated by this box here, seem to have a boron isotope composition most similar to borate. And that suggests that borate is the predominant form of boron in solution that's taken up by the carbonate. What this means is that you can make boron isotope measurements on carbonates and relate it back to the borate relationship with the solution pH and effectively calculate back the pH of solution that the carbonate mineral formed from. So this is some of our initial boron isotope data on different marine carbonates from Justin Ree's 2009 experiment. And the first thing to note is that we measured quite a range in boron isotope compositions between different species. And the next thing to note is that we can take these measurements, boron isotopes, trace them across to this borate pH relationship, trace them down and infer the pH of the calcifying fluid. What this data suggests is that the pH of the calcifying fluid of this coral species that was cultured is significantly higher than the calcifying fluid pH of this oyster on the other end. And when we look back to Justin's experiment, we found that this coral was relatively refractory to the effects of high CO2 conditions. It was not affected until it reached the very high CO2 conditions, whereas this oyster showed a negative response, so it was very sensitive to elevated CO2 conditions. And so our boron isotope data is entirely consistent with the ideas proposed in that conceptual model, that the coral is actively regulating the pH of its calcifying fluid, but the oyster is not. So you can see here that the inferred pH of the calcifying fluid from the boron isotope measurement is more or less exactly the same as the pH of the seawater in which it grew.
Boron isotope measurement gave you instantaneous signal or lifetime for the organic?
So you have to, so in this experiment we're specifically sampling new growth that occurred over the course of the experiment.
Just for new growth, not during the lifetime of organism?
Yeah, well, I mean, you know, a coral will put on new growth over its lifetime. You can potentially make boron isotope measurements spanning its entire lifespan as it accretes its skeleton, yeah.
But in this experiment, you were just doing
the new growth in the new culture conditions, yeah. So some data here from kind of tropical warm water coral species grown at varying CO2. And here, in both these species, we saw a drop in the pH of calcifying fluid, as CO2 was increased. And taking this back to our conceptual model, this would be consistent with a kind of linear negative calcification response. Carbonate ion consistently dropping in response to elevated CO2 conditions in these shallow water tropical species. Yeah.
Question about refractory or threshold behavior.
Mm-hmm.
I think you said it in words, but that's an organism you're assuming that is able to adjust the proton pump to keep conditions ideal up until a certain point. Is that how I understand it?
Yeah, I mean, that would be the implication, yeah. So our work in this area is really focused on kind of three...
Have you found any examples yet of such an organism that you can analyze in this way?
of a threshold response.
A refractory interval.
Yeah, we have.
Regulating its proton pump.
Yeah, we have actually one coral species where the boron isotope composition seemed to be relatively invariant. I didn't present that because we want to go back and repeat that and make sure it's a real result. You know, if you see no change in your experiment, you want to make sure you did the experiment right. But yeah, we have seen an organism where there's no change over the culture conditions. Yeah, so our work in this kind of area is focused on three main foci. One is to utilize this array of different culture marine organisms from the 2009 paper and use our geochemical toolkit to try and understand the diversity in responses. We have a new funded project that's going to involve new cultures where we're going to take a suite of different corals that we're hypothesizing are going to be different in their ability to pump protons away. Maybe they grow in very different environments that have different saturation states today, so we would hypothesize they might be different in their capacity to regulate internal pH. What we're going to try and do in that case is combine the geochemical analysis with the microelectrode and pH-sensitive dye approach all in the same cultures to see if we can get self-consistent results. And the next project, which is the project of a postdoc of mine in France at the moment, is to apply these same ideas to calcifying phytoplankton, coccolithophores. These are the organisms that are potentially relevant for global biogeochemical cycles. Just a bit of background on this. So our understanding of the impact of OA on coccolithophores has changed somewhat over time. So for a while, this 2000 paper was a kind of gold standard in this field. In this paper, they grew a couple of different species of calcifying phytoplankton at varying pH, and they saw negative calcification responses to changing pH. They also saw that low pH cultures, kind of aberrant morphologies in these cultures. And a crucial thing to note, though, experiments from this type of, from this era of OA experiments typically manipulated the pH of their cultures using kind of acid and base titration, so not bubbling CO2 through the culture. But there was a major reassessment of this in 2008 in a paper published by one of my collaborators, Deborah Iglesias-Rodriguez, in experiments where she was manipulating the oceanic carbonate system and pH by bubbling CO2 through the cultures. In the same species of coccolithophore, she actually saw a positive calcification response. In other words, they were producing more carbonate as CO2 was increased in this experiment. This was initially interpreted as being to do with the difference in experimental setup. It relates back to the kind of conceptual model about the input of DIC into the system that I was just talking about. It's become better appreciated since then, however, that probably also a factor in this is genetic variability in E. huxleyi. So this study used a genetically different strain of E. huxleyi to previous studies, and it's now been shown that there's a huge genetic diversity in this phytoplankton, both within populations from ocean basins, but particularly there's a lot of diversity between populations in different ocean basins that influence its response to high CO2 conditions. And so to try and understand the response of calcifying phytoplankton to CO2, it's important to recognize that their system of calcification is somewhat different from the systematic that I showed you earlier. They actually have their own subcellular compartment, the coccolith vesicle, that is devoted to generating these plates that go on the exterior of the phytoplankton. And there's very strong biological control over the import of calcium and bicarbonate into the coccolith vesicle. They're also unusual in that they actively import bicarbonate into this vesicle to facilitate calcification. So that's a significant difference from that schematic that I was describing to you earlier. And this is kind of very new data. It's only recently been generated by my postdoc in France. But Yiwei Liu has basically been using this boron isotope approach to estimate the pH of the coccolith vesicle in this one species of Coccolithophore. This is really the first geochemical determination of the pH of the coccolith vesicle. It's not really, because these are so tiny organisms, it's not really possible to use any of the other approaches to do this that I was mentioning. The first thing she noted was that the coccolith vesicle pH is not significantly elevated above that of seawater. And an interesting finding was that an experiment where two different strains that were isolated from two different ocean basins, she found that one of these strains showed a clear relationship between coccolith vesicle pH and ocean acidification caused by high CO2, here plotted as bicarbonate ion concentration. But the other strain that she examined didn't show any response to OA. So again, clear evidence of genetic differences influencing the response of phytoplankton to high CO2 conditions. And so this is an area we're planning to explore in a lot more detail, and another researcher working with me, Jill Sutton, is in the process of developing a kind of culture apparatus, not just to culture these calcifying phytoplankton over a short term, but really to culture them over long-term experiments to generate maybe 400 to 1,000 generations at high CO2 or a variable temperature. These experiments would run for 16 to 18 months potentially, and the interest here is that you can potentially then look to characterize adaptive responses relating to the fact that populations may have, maybe genetically favorable to generate a positive versus adaptive versus negative response to ocean acidification. So this is something we're setting up at the moment. We're going to look at these adaptive cultures with our geochemical toolkits. We're also kind of in discussion about potentially looking at functional genomics works with these cultures, transcriptomics, proteomics maybe, to try and understand what the molecular basis for these potential adaptive responses are. So this is something we're hoping to focus on in the near future.
Quick question, will you be using a strain? I mean, there was a study showing this evolutionary adaptation to high CO2 by India. I think that was India actually. Are you using the same strain and then
Yeah, I mean, we're hoping to do both things, really. We're hoping to take different strains isolated from different ocean basins and compare them. So that'll be something that's not been done before. But we're also hoping to take genetically mixed isolates taken from the field and also do these kind of long-term adaptive responses. So we're hopefully going to do both of those things. And the kind of, this is an initial experiment. You can also imagine adapting this experiment to also look at the kind of combined effects of CO2, variable temperature and nutrients, for example. And so an area that we're not currently pursuing, but I think would be a strategic area to consider pursuing, particularly here in LA in the context of the AltaSea initiative, is developing facilities to do larger scale and more complex culturing experiments. And one potential justification for this is the idea that in different environments have very different variability in pH and oceanic conditions that they experience in the kind of natural cycles. By this, so here on the left here, I'm plotting some data from a paper from Gretchen Hoffman, where she took a month-long time series of oceanic pH in a variety of different sites. She found in the open ocean that in the course of a month, pH doesn't vary very much. But in coastal sites, coastal upwelling sites off the coast of California, you can see there's substantial variation in pH over the course of a month, and also in a kelp forest environment here. And so this begs the question, to me at least, what effect is an anthropogenic pH change going to have on top of an environment that undergoes such large pH changes anyway? Presumably the marine calcifying organisms that live in these environments are somewhat adapted to resist these kind of changes. And so what effect would a kind of 0.1 or 0.2 extra acidification have on species that are already living in these very variable environments? And that's the sort of target that you can imagine having kind of more complex culturing facilities would allow you to address those sort of questions.
What can you do at AltaSea that you can't do here?
So you would need a kind of larger scale culture apparatus with like seawater being pumped in, perhaps. You would need sort of complex regulation of the water that was being brought into the culture environments. That would be perhaps a little bit difficult to set up in countries. So that's a kind of summary of what we're currently working on. The kind of questions that I think are important to expand on in this area and perhaps focus on in future work how multiple stresses like temperature and hypoxia potentially compound the effects of ocean acidification on different marine organisms. This question of whether some species are able to adapt over time, and can we characterize that in long-term culture experiments, I think is very important. And is it possible to generate data from these kind of culturing controlled experiments that's actually relevant or useful for modelers and biogeochemical models? And so just in conclusion, I hope I've convinced you that by kind of combining techniques from geochemistry and cell biology, we can get new insights into the response of organisms to their physical environment. We've generated data that shows that there's quite a wide variation in organisms' ability to regulate their pH and carbonate system of their internal calcifying fluid, and ultimately that may take us some way towards an explanation for why marine organisms show such diversity in their response to OA. And so I'll just thank my collaborators. I'll point out that we've had a number of UCLA undergrads working on this, supported by the SACNAS and CARE program, and research programs currently funded by both NSF but also LabX at the European Institute of Marine Science. So thanks for listening, and I'll take any questions that you might have.
So with the long-term culturing study, are you going to have just different cultures set at specific CO2 levels, or are you going to be gradually increasing the CO2 levels over the duration of the entire experiment?
So the way we're setting it up at the moment is we'll have 4 incubators at 4 different CO2 conditions, and we'll probably put a series of kind of flasks in there that might contain different strains, different species that can be run concurrently. There's some interesting questions associated with if you acclimatize or allow a culture to adapt, you could then take it from one regime and put it in another. That's another whole set of questions that you could potentially ask that we're also thinking about. So if you allow a culture to adapt in CO2 and then expose it to a higher CO2 condition, is it more able to cope with the higher CO2 after it's adapted to some extent? So
it seems like if it's possible to have one culture where you're gradually increasing it over time, it'd be a really useful comparison to go gradually from the lowest levels to the highest levels and compare the differences, because as people are doing these types of experiments, the environment's not changing in a stepwise fashion.
Right.
And so, we're missing a lot of adaptive potential.
yeah, that's probably a very good point. Yeah, that's a good point.
David?
Could you speculate for a moment on the steps that would be required to go between the organismic level experiment and the biogeochemical cycle, presumably identifying key parameters that would be needed, which need constraint biogeochemical, build biogeochemical label models of this, and saying something about it inferred from the organismic level, or what's the, so taking it back to something you said in your introduction, essentially.
Yeah, I mean, I think I'd like to imagine that an experiment where you could constrain how the rates of inorganic carbon production in key primary producers might change, and an adaptive response to CO2 in culture over time. That would give you maybe some constraints on a biogeochemical model to say, well, if we're simulating change in primary production or inorganic carbon production in the upper ocean, this is perhaps how it's likely to change over time based on our long-term culture experiment.
Growth rate parameters are often the key translation of organisms transformation. Can you imagine getting at that in the experiment?
Well, the growth rate of these phytoplankton in culture is something that's kind of routinely measured as part of the experiment, so definitely, yeah.
In terms of the more alarmist scenarios that a lot of people are coming up with about ocean acidification, everything is going to go extinct, do you think that the fossil record is relevant? What happened at the end of the Permian, the transition from the Triassic to the Jurassic and so on, where there were major changes in ocean pH and CO2 levels, are those likely to be relevant?
Well,
things did survive, and obviously we have all these organisms now.
Well, I think it's very, I'm personally quite interested in historical geological ocean acidification events. Of course, it's debated how useful analogues those really are, because they generally happened over much shorter timescales than we're seeing at the moment. So an organism's ability to adapt and evolve in response to those types of acidification events, they have a much longer time period to do so than organisms in the ocean will do today.
Yeah.
Can you think of a model metazoan organism that has like a shorter generation time to be used as an evolutionary study? And that also is kind of easy to keep. I'm just thinking there's most of them are more longer.
Yeah, exactly. Well, I mean, the reason for focusing on coccolithophores for that experiment is that they have such short generation time, so I'm not quite sure whether there is an easy experiment that you could think of.
Do you actually know if the coccolithophores with the chitin have any effect or show any effect?
Yeah, I mean, I think there's much less literature on that, and I think that at least the initial experiment suggested that there wasn't a significant deleterious effect on copepods. But I think, there's just not a lot of literature on it, so yeah. Maybe kind of the sort of behavioural and, tissue-related ionic imbalances might be more relevant to those sort of organisms than actually like the biomineralization aspect.
Well, Bill, why don't you go ahead?
So you mentioned genetic diversity in the E. huxleyi response to pH. Did it correlate with the geographic diversity and the amount in the CO2 signal in those different ocean basins?
The genetic diversity, did it? Sorry, can you repeat the question?
Population differences between the E. huxleyi and the response to carbon dioxide challenge, and
yeah.
Did it correlate with the actual oceanography of those basins? Were there?
That's a good question. I don't think there's not an obvious correlation in what we've done so far. I would be surprised if there wasn't, though, when people look at it more closely. Like it would make sense if, coccolithophores growing in the high Arctic would be somewhat differently adapted to those growing elsewhere in higher saturated waters. I don't think, in our experiments, we've not done enough work to see that, but that's like something that we're hoping to explore.
Okay, let's thank our speaker one more time.